Puberty in animals is not just about age, it depends on nutrition, metabolism, hormones, and the environment. In this video, we explain how these factors work together to control the onset of puberty in females. A certain degree of body fat is required for the onset of puberty in females. At birth, the primary priority of the neonate is survival, and most available energy is directed toward maintaining vital physiological functions. Reproduction is non-essential at this stage and remains inactive. As the young animal grows, body mass increases, surface area relative to body size decreases, and metabolic efficiency improves. Energy expenditure then extends beyond survival needs. As metabolic rate gradually decreases, excess energy becomes available and is stored as fat. Once sufficient energy reserves are established, the body can prioritize reproductive development and puberty begins. However, the exact fat threshold required to initiate puberty has not been precisely determined. Hypothalamic neurons that regulate GnRH secretion can detect moment-to-moment changes in circulating metabolic fuels such as glucose and free fatty acids. A central question in reproductive physiology is how metabolic status activates GnRH neurons. Research shows that high-frequency GnRH pulse generation is influenced by blood glucose and fatty acid concentrations. Experimental studies in female hamsters demonstrate that blocking glucose and fatty acid oxidation disrupts estrous cycles by impairing GnRH secretion. These findings indicate that hypothalamic GnRH neurons are sensitive to nutritional and energetic status. The effect of nutrition on puberty is clearly seen in dairy heifers. A main goal in dairy farming is for heifers to calve by 24 months of age, which requires proper nutrition and adequate body growth. Heifers fed high levels of nutrition early in life grow rapidly and reach puberty earlier, sometimes as early as 6 to 8 months. However, excessive feeding can lead to overconditioning. Heifers fed a moderate, balanced diet grow steadily and reach puberty slightly later but at the correct body size. In contrast, heifers fed poor quality or restricted diets grow slowly, reach puberty late, or reach puberty at an inadequate body size, making them poor candidates for pregnancy. Thus, nutrition affects both the timing of puberty and reproductive success. Any discussion of metabolic control of puberty must include leptin. Leptin is a peptide hormone secreted by adipocytes, and its circulating concentration reflects body fat content. Leptin receptors are present in the hypothalamus and anterior pituitary. Leptin is considered a signal that informs the brain that sufficient energy reserves are available. It likely acts as a permissive factor that allows hypothalamic neurons regulating GnRH secretion to activate once adequate body fat is achieved. Kisspeptin neurons appear to play a central role in this process. These neurons project into hypothalamic regions rich in GnRH neurons, suggesting direct interaction. Metabolic signals such as leptin, glucose, and fatty acids are thought to stimulate kisspeptin neurons, which then activate GnRH neurons and promote pubertal onset. However, this model remains hypothetical and has not yet been conclusively proven. Environmental and social conditions also influence pubertal onset in females. External cues such as season of birth, photoperiod, and social environment are detected through sensory systems, mainly optic and olfactory pathways. These signals are processed by the central nervous system and influence hypothalamic GnRH neurons. When metabolic conditions are optimal, these cues can advance high-frequency GnRH pulse generation. Season and photoperiod are especially important in seasonal breeders because changes in day length are translated into hormonal signals by melatonin, a hormone produced by the pineal gland during darkness, at night. Melatonin acts as the key messenger between the environment and the reproductive axis and stimulates the hypothalamic, pituitary, gonadal axis and promotes puberty. In sheep, which are short-day breeders, spring-born lambs experience increasing night length in autumn, lead to increased melatonin secretion, allowing puberty at about 5 to 6 months of age. In contrast, autumn-born lambs grow during increasing day length, which reduces melatonin secretion, suppresses reproductive activity, and delays puberty until the following fall at about 10 to 12 months of age. In cattle, heifers born in autumn often reach puberty earlier than those born in spring, suggesting a milder photoperiod melatonin influence. In dogs, puberty shows minimal seasonal influence, indicating limited melatonin involvement. In cats, which are long-day breeders, decreasing melatonin secretion during increasing day length strongly promotes pubertal onset. All seasonal effects of melatonin on puberty depend on adequate nutrition and normal growth. Social cues also play a major role in pubertal timing. Olfactory signals, particularly pheromones in urine, can accelerate or delay puberty. The presence of a male hastens pubertal onset in species such as sheep, pigs, and cattle, providing an evolutionary advantage by improving reproductive success. However, social stimulation cannot override inadequate metabolic status. Animals must still achieve sufficient body size and energy balance. Certain social conditions can delay puberty. In swine, gilts housed in small groups reach puberty later than those in larger groups. Exposure to a boar can counteract this delay, accelerating puberty regardless of group size. Both visual and physical contact are effective. Similar effects occur in cattle, where exposure to bulls accelerates puberty in beef heifers, especially when combined with higher growth rates. Compared to females, the role of metabolic status in male pubertal onset is poorly understood. This is likely because the energetic cost of male reproduction is much lower than that of pregnancy, parturition, and lactation in females. In addition, few studies have examined social influences on male puberty, as most research focuses on male effects on females. Thanks for watching Vetcare. If you found this helpful, don't forget to like, share, and subscribe for more simple and clear veterinary lessons.